The heart is a muscle that contracts to pump blood throughout the body. It contracts independently from nervous system input through the mechanisms of pacemaker cells and cardiac myocytes. An action potential is generated by pacemaker cells that are found in the sinoatrial node. They spontaneously depolarize to generate an action potential that is then propagated throughout the successive steps of the conduction pathway. Cardiac myocytes conduct electric impulses in order to propagate the action potential through the rest of the heart. The action potential first travels through the atria to cause them to contract simultaneously. This is the first part of the heartbeat. The action potential then travels to the atrioventricular node, extends throughout the Purkinje fibers and into the ventricular myocytes to make the ventricles contract. This causes the second part of the heartbeat. The sinoatrial node, also known as the SA node, is the primary pacemaker of the heart. It initiates all heartbeats and regulates heart rate. If it fails, other parts of the conduction pathway then have to spontaneously depolarize to generate this action potential. In order for the action potential to move between cardiac cells, myocytes are connected by gap junctions, which form channels that allow ions to flow from one cell to the other. An action potential in one cell will trigger an action potential in its neighbor, and the signal propagates rapidly. Action potential generation and conduction are essential for all myocytes to act in sync. Looking more closely at the SA node, we zoom into the molecular level. Here we see a typical pacemaker cell. The membrane separates the contents of the cell from the extracellular environment. More importantly, the charges are kept very different on opposite sides of the membrane in order to create a very regulated environment for when the action potential begins. The extracellular environment is positive and naturally has many calcium and sodium ions. The intracellular environment is negative and has a much smaller concentration of ions, which are mainly potassium. This difference in concentration is called a concentration gradient, and it helps drive the changes in membrane potential throughout the process of depolarization. Within the membrane, we see multiple different channels, such as the L-type calcium channel, T-type calcium channel, the funny channel, and the potassium channel. At different points throughout the action potential, these channels open and close to allow the flow of ions across the membrane. At rest, the membrane potential is negative 60 millivolts. This can be seen on the graph in the upper right corner. Pacemaker cells are unique in their ability to spontaneously depolarize. This is why they are responsible for pacing the heart. The spontaneity is driven by the random opening of funny channels, also known as HCN channels, which open to allow small influxes of sodium. The influx of sodium brings some positive charge into the cell to help the membrane potential drift from negative 60 millivolts towards a more positive potential. As it passes negative 50 millivolts, the T-type calcium channels begin to open and allow calcium to enter the cell. These channels are transient channels, meaning they open slowly. The early influx of calcium does not cause depolarization. It just helps to further drive the membrane potential to be more positive. As the influx of T-type calcium helps further depolarize the cell, the membrane potential continues to rise until it reaches negative 40 millivolts. This is known as the threshold for depolarization. At this point, the L-type calcium channels now open, and a large influx of calcium rushes into the cell. This significant rise in positive charges is what causes the cell to depolarize. Around the same time that L-type calcium channels open, potassium channels begin to open. However, these channels open relatively slowly and only allow for a small amount of potassium to exit the cell at a time. This allows the cell to gradually repolarize until the membrane potential goes back to its resting state at negative 60 millivolts. Once this has been reached, the cell is repolarized and ready to generate another action potential. As the action potentials are generated in the SA node, they then spread through the conduction pathway to activate the subsequent contractile myocytes. Similarly to the SA node, there is a membrane potential in cardiac myocyte cells due to the different concentration of ions opposite the membrane. Once again, sodium and calcium are in high concentration in the extracellular space, and potassium is in higher concentration inside the cell. However, there is a much higher concentration of positive charges outside the cell than inside, and the resting membrane potential here is negative 80 millivolts. By looking at the membrane, we can see that a ventricular myocyte have some of the same channels as seen in the SA node, but there are a few new ones, including the sodium-calcium exchanger and the ryanodine receptor. It is important to note that the sodium channel is no longer a funny channel, because those channels are unique to the SA node. This means it will not spontaneously depolarize like it did in the pacemaker cell. Instead, in a cardiac myocyte, an action potential begins with receiving electric stimulation from the previous cell in the pathway. As mentioned earlier, the action potential travels through the gap junction between cells, and a rush of positive charge will locally accumulate in the extracellular space. This will activate the sodium channel and cause a very fast influx of sodium. As this happens, we see a huge spike in the membrane potential as it rises right up to 40 millivolts with a sudden increase in positive charge. This marks the depolarization of the ventricular myocyte. After depolarization was achieved via the sodium influx, we now see this interesting plateau period where the cell isn't really depolarizing, but it isn't repolarizing yet either. This is maintained by the sodium-calcium exchanger and the slow influx of calcium through L-type channels. The sodium-calcium exchanger pumps out 3 sodium ions as it pumps in 1 calcium ion, and the constant move of charges across the membrane is what allows for the plateau period to occur. While all that was happening, as the membrane potential passed negative 40 millivolts upon depolarization, L-type calcium channels opened and began to implement a slow and steady influx of calcium. Additionally, as calcium is entering the cell through L-type channels, it goes straight to the ryanodine receptor and binds to it. This can be seen in the lower right-hand corner. This causes ryanodine receptor to release a wave of calcium from the sarcoplasmic reticulum. The sarcoplasmic reticulum is a membrane-bound organelle within the cell that contains huge amounts of calcium stores. It is this release of calcium that subsequently causes the ventricles to contract. Finally, repolarization occurs as potassium channels drive the efflux of potassium and the membrane potential returns to its resting state.